Now, the disease itself and the pathogenesis of it, as I alluded to before, the virus attacks predominantly the CD4 positive T lymphocytes, and they're our T helper cells and largely responsible for control of the immune system. There's a bit of a difference here between HIV and FIV in the way that the virus actually gains entry to the cell. For FIV, or our cat AIDS, it binds to the CD134 cell surface antigen, and then will bind additionally to the CXCR4 cell surface antigen receptor, uses the N protein of the virus, and once the protein is bound to those two receptors, it induces a conformational change which exposes the fusion region of the protein, and that then allows the virus to fuse with and enter the cell. Once the virus has entered the cell, the viral reverse transcriptase will create DNA from the viral RNA, and that's then incorporated into the cellular DNA after it's been transported into the cell nucleus. Cunning little virus then decides to use the cellular machinery to create more virus, and on the cycle goes. An important point about lentiviruses in general, and FIV as well, is that viral reverse transcriptase is an inaccurate enzyme and has no error correction ability. At first glance, that would seem to be a disadvantage to the virus, but it turns out it's very advantageous. And certainly there are a large number of defective copies of the virus made, but it's what allows the virus to mutate rapidly and change rapidly and perhaps escape immune suppression or escape drug therapy or difficult to vaccinate and so on against, is because of this inaccurate non-error correcting reverse transcriptase that it uses.